So in this recording, we want to address the topic of mRNA processing. And this is where we're really going to see a huge divergence between prokaryotes and eukaryotes. So I'm going to mention prokaryotes now, and then I'm pretty much going to leave them behind, because the rest of this information about mRNA processing is going to be focused on eukaryotes. In prokaryotes, bacteria, the primary transcript, i.e. that mRNA molecule that is built from the template DNA, is the messenger mRNA. That's it. It's ready to go. All right. Now, in eukaryotes, that primary transcript, that copy of the mRNA of the DNA molecule, in fact, is a pre-mRNA. And we're going to see modifications need to be made to both the 5' and 3' ends, and then we're going to have to remove what are called introns, or non-coding sequences, from the pre-mRNA before we have a maturation event of that mRNA and it's ready to be translated. All right. So the alteration of the mRNA ends involve a 5' cap and a poly A tail, and these are going to play a role in protecting the mRNA, determining how long it lasts in the cytoplasm, facilitating its export, and how it interacts with the ribosome. So if we look at this picture of that mRNA, we can see we have our start codon and our stop codon, the protein coding segments. So upstream, or 5' to the start codon, you see a 5' UTR. That's a 5' untranslated region. 5' to that, you see what's called a 5' cap. Now, 3', or downstream from the stop codon, you have a 3' untranslated region, and a signal in there that says polyadenylation. So this is now going to be the information to say, hey, after me, I want you to add a bunch of A's onto the end of this mRNA. And that's called a poly A tail. And so these are our modifications that we need to make to our mRNA to facilitate its functioning properly in the eukaryotic cell. So if we look at that 5' cap, it's interesting chemistry, right? So now what you've actually done is made a 5' to 5' bond. So you've kind of broken the rule that we always build 5' to 3', but this is going to be different. It's not encoding information. It's going to actually make this little kind of nub at the end of the mRNA, which we believe has a role in helping it to bind to the ribosome. Now, this seems like a whole lot of extra work, but it's necessary in this complex system. Now, the real meat of RNA processing, though, comes in the removal of introns. So this is called RNA splicing. And so splicing is a general term, like if you splice wire, you cut it, and then you put it back together, right? You may have added a section or put in a different connection, but we're going to splice our RNA. To do that, we're going to need a whole bunch of machinery that's going to remove the introns. So this is where we need to introduce our new term, and this is the source of eukaryotes being much more genetically complex than prokaryotes. So in your DNA, in your genes, when we make that primary transcript, we actually have regions that encode for amino acid sequence and regions that do not. So the regions that encode for amino acid sequence are called exons, and regions that do not are called introns. So you want to think about introns being inserted. They interrupt the amino acid sequence. And so they're displayed here as light pink, and those need to be removed, because if you try to translate them, you're going to get gobbledygook. You're going to get a protein that's junk. It's not going to work. All right? So what we have here is the machinery is called the spliceosome. And it's going to consist of a variety of proteins, and again, proteins that are an RNA protein hybrid. So these small nuclear ribonucleoproteins are a combination of protein and RNA. And that's going to be important for recognizing the RNA that needs to be processed and catalyzing the reaction to cut it open. So here is an example of splicing. So we have exon 1 separated from exon 2 by an intron. And so this complex, the spliceosome, is going to assemble. It recognizes the borders of the intron and then literally snips them out and then splices together exon 1 with exon 2. Again, compared to a prokaryotic system, this seems like a whole lot of extra work. Why would you do this? Why would this be necessary when efficiency seems to be so important in biology? Well, it actually, in the long run, is really, really efficient, because this is the source of getting more than one protein from a single gene. Because I'm splicing the RNA, removing introns, it gives me an opportunity to do this differentially. So I have alternative RNA splicing. I can splice out different introns and different exons and ultimately create different mRNAs from the same gene. Right? And that's going to be super important in creating more complexity, the complexity that we see in eukaryotes versus prokaryotes. So in this picture, what we see here is our gene, and then our mRNA copy of the gene. Now the boxes here represent the exons. Now alternative splicing says that when this engages the spliceosome, this could result in protein A or protein B. Protein A is exon 1, 2, and 3 spliced together, while protein B is exon 1, 2, and 4 spliced together. These are different proteins from the same gene. Now, a lot of the pictures that you're going to see of introns and exons as we look at gene structure in eukaryotes are going to show you these big exons with small interrupting introns. When this is a picture from a research paper which shows you the map of a gene, and what you can see here is the boxes representing the exons and the lines representing the introns. And so if you just look at A, for example, you can see that you have non-coding sequence, and then you have 33 nucleotides of exon, of coding sequence, followed by 726 nucleotides of intron, of non-coding sequence. So those 33 nucleotides need to be spliced to the following exon that's 204 nucleotides. And that's followed by an intron that's 5.6 kilobases. It's 5,676 base pairs, or nucleotides, of non-coding sequence that will need to be spliced out. So you can see that the structure here can be relatively complex, and that's going to, again, add to the diversity that can be created from a single gene. In humans, the average, the last I read, was 8.7 different proteins from every gene that you possess. Right? Now, there oftentimes is some structure to this shuffling, and we think about proteins working oftentimes in domains. So these are regions of a protein that actually result in a certain function of the protein. And oftentimes exons or groupings of exons will correlate to protein domains. So what we see here is that domain 1, 2, and 3 correspond to exon 1, 2, and 3. And each of those domains, you want to think about them as kind of a tool on a Swiss Army knife. So if the protein is the Swiss Army knife, domain 1 may be the knife. Domain 2 may be a saw. Domain 3 may be a corkscrew. And they're all packaged nice and neatly and efficiently together, but if you separate them, they could work to a certain extent on their own. And depending on the protein, that can or cannot be true. And this is a vast generalization, but it's the starting point to help us understand, again, the diversity of eukaryotic genetic complexity and how proteins ultimately work.